Monday, and then we'll and
We'll see. In
um,
the future, I'll look at these guys.
today, let's start here with the model of tumor genesis that originally was proposed in the 70s, long before any of the genes were identified, but still holds. Basically, you think of cancer as waves of clonal evolution. Pretend those green dots are epithelial cells in the colon. One of those cells acquires a mutation, somatic. Somatic, everybody knows what somatic means? Okay, not in the germline. And that cell has a selective growth advantage. Selective growth advantage allows it to proliferate, which I'll explain in a bit more, than the sister cells, the normal colon epithelial cells. Those cells then divide, they become a small clonal population. One of the progeny acquires another mutation, so now there's two mutations indicated in blue. That cell has yet an additional selective growth advantage over its sister cells, expands once more in a second wave of clonal expansion, and this process continues to occur throughout the lifetime of the tumor. In general, as we discussed on Monday, it takes approximately 3 waves of clonal expansion to get to a cancer. That is a typical epithelial cancer of adults. It takes only 2, roughly, again, to get to a liquid tumor, leukemia, lymphoma, or some pediatric tumors. Although this process never stops, these mutations, of course, continue to occur in cells of the tumor, as well as in cells in normal cells in everyone. And if you treat a patient with cancer, some of the mutations that have been acquired during the evolution of the tumor can give resistance or endow the tumor, that cell with the mutation, with resistance to the therapy. And that basically explains why cancer therapies don't work. When you're treating patients with advanced cancer who have literally billions of cells in their tumors, there are many cells which have mutations which confer resistance to any therapy you throw at it. So even though most of the cells in a good therapy may disappear, it's only a matter of time, fait accompli, until those cells grow out to yet another wave of clonal expansion after the cancer is formed, after the treatment is administered, and the patient will relapse. And it's just more of the same. You can think of these 3 waves in more discrete terms. Whoops. The first wave can be considered tumor initiation. There are 3 stages of tumor genesis. Tumor initiation, that is a result of the first mutation. The second mutation endows a selective growth advantage, which will allow the tumor to progress. That means it simply increases its size, the number of cancer cells. And then the third mutation is the straw that breaks the camel's back, so to speak. It not only endows a selective growth advantage, but it endows a selective growth advantage in a particular microenvironment. And that environment is what we see as invasion. Normally, cells are very well regulated. They can't cross the basement membrane, and if they do, they won't survive because they need cell-cell contacts. They need particular cytokines or other constituents in order to grow. But cancer cells which have these mutations can cross the basement membrane either by accident, when there's a hole in the basement membrane, or on purpose if they acquire expression of some enzymes like proteases that allow them to traverse the basement membrane, and then they'll grow there. Moreover, they can continue to expand, to invade the underlying tissues, get into a blood vessel or lymphatic, travel to the liver or to the lung. Normal cells in that environment will not grow. In order to be a properly regulated multicellular organism, unlike an amoeba or a single-cell organism, the cells have to regulate themselves extremely carefully. And if they don't, they get a cancer. That's what we call it. So if a normal cell gets trapped in the lung, for example, in a capillary bed in the lung, it will not survive. It certainly will not grow. It doesn't have the cell-cell contacts, all the other biochemical signals that it needs to proliferate. It'll simply either die or senesce. But a cancer cell, by virtue of these additional somatic mutations, can survive there. That cell can proliferate and then over the course of a couple of years become a visible metastasis containing 100 million or billion cells. Let's look for a few minutes at the nature and number of mutations that occur in a typical tumor. This could be a colon cancer, pancreatic cancer, breast cancer, esophageal cancer. It doesn't matter. The principle is the same. Here, there are 23 chromosomes organized like a piano keyboard, a strange one, but kind of like a piano keyboard. And as we discussed on Monday, there are roughly 50 to 100 mutations indicated by those round tires on this diagram. Now, most of those mutations are not important. They're called passengers. They just have accumulated as cells have divided. Remember, every time a cell divides, it acquires 3 to 5 somatic mutations. And here we're only looking at coding mutations. And in fact, those that alter the amino acid sequence of a gene, so there are only 50 to 100 of them per cancer. Now, of those, let's say there are 50, there are only 3 or 4 that actually drive the process of tumorigenesis. The rest are simply passengers. And when I say drive, I'll explain that in a few minutes. But those genes are either oncogenes or tumor suppressor genes. The oncogenes can be thought of as accelerators in a car. If you normally press on the accelerator, the car goes. If you have a broken accelerator that's stuck to the floor, the car will continue to go. And similarly, an activated oncogene will continue to impart its selective growth advantage and make the cells grow when they ordinarily should stop because of the signals communicated to them both in the milieu, that is the humoral signals, as well as the intercellular signals from cell contact. Suppressor genes are the brakes. they normally signal cells to stop growing. If you have a dysfunctional break, then the cell won't stop even though it should. And to get to a cancer, it generally requires 3 broken parts. 1 oncogene and 2 tumor suppressor genes, or 3 oncogenes or 3 tumor suppressor genes, doesn't matter. And it's just like having an accelerator, a foot pedal, and an emergency brake. It takes all of those broken things for you to prevent stopping the car when you're driving. And similarly, this is a backup to keep cells from getting cancers. A third class of cancer-causing genes. Remember, all these genes individually contribute to cancer, but in aggregate actually cause cancer, are the repair genes. These are genes involved in the repair of damage that occurs every time cells divide. They make mistakes. So the polymerases themselves are error prone. They make errors just by the fact that they are not perfect replicators of DNA. So, Lauren, tell us why in 5 billion years of evolution, these polymerases have not evolved to become perfect copy machines. They make mistakes.
Does it have to do with like creating more genetic diversity by increasing mutations?
Basically, if they didn't make mistakes, we wouldn't be in this room, right? We would be some sort of proto-organism, a bacteria or something, right? So, cancer, when we look at it that way, is a side effect of evolution. You have to make mistakes. Now, the polymerases themselves make lots of mistakes. Remember Haley, what's the rate of mutation in normal cells and cancer cells? Do you remember that from last time?
I don't remember. I don't know if I wrote it down.
Well, remember, order of magnitude is roughly 1 in a billion mutations per base pair per cell division, right? But that's the rate of mutation after the repair systems have fixed up the genome and corrected all these mistakes that polymerases have made. The actual rate of mutation of the polymerases is 100 times higher than that, 100 to 1,000 times. So perhaps 1 in a million mutations per base pair per cell division. So these repair systems, and there's several, are extremely important for limiting mutations. If one of them becomes inactivated, that leads to the acquisition of mutations at a much greater rate, and that will include all mutations, all genes in the genome, but including oncogenes and tumor suppressor genes. So these repair genes contribute to cancer not in the same way as oncogenes or tumor suppressor genes, not through a direct effect, but rather through an indirect way, that is increasing mutation rate. They act similar to cigarette smoke or ultraviolet light, increasing mutation rate, enhancing or accelerating the process of tumorigenesis. And certain patients who have mutations, who inherit mutations in repair genes, are prone to develop cancer for that reason. And good examples are the BRCA1 and BRCA2 genes, which predispose to breast cancer and ovarian cancer, as we discussed last time. They do that through suppressing changes in recombination, structural changes to the genome, mainly. And another major class of repair genes is called mismatch repair genes, which, when inherited in a mutant form, can cause another type of predisposition, which contributes to colorectal cancer, stomach cancer, uterine cancer, and several others. So these repair genes play an important role. Now, what do they do? Again, we're taking the 40,000-foot view here. You don't need to remember the names of the genes, but I hope you remember some of these high-level principles. They all do the same thing. Normally, in normal adult cells, the rate of cell birth precisely equals the rate of cell death. For every cell that's born, that cell dies, or its progeny dies. And this rate, the net cell growth, is dependent on the difference between cell birth and cell death. Now, in evolutionary genetic terms, selection, defined evolutionary in evolutionary biology, but we also use it in cancer genetics, is this difference, selective cell growth. So in normal adult cells, Valeria, what is net cell growth? So I have that right, Valeria?
Mm-hmm.
What is net cell growth in all of your cells and every other adult on the planet who is functioning normally?
As in a number?
Pardon? Yeah, a number. Precise number.
Well, if you said that cell birth and cell death equal each other, typically, so net cell growth would be...
You're on the right track.
0?
0, correct. It's precisely 0. Suppose it's less than 0. There's more cell death than cell birth. What happens? Tissues atrophy. This is what happens with aging. This is why old people's skin, like mine, gets so thin. Doesn't have enough cells anymore. It atrophies. The opposite. If there's more cell birth than cell death, that's neoplasia. Except in injury, right? When a tissue is injured, you'll have to recover those cells. So that's a normal physiologic process. And some of the pathways in cancer have developed from what organisms need to do when wounded to, under certain circumstances, be able to renew their cells and have a net cell growth that's greater than 0. But in the absence of injury, this defines physiologically what neoplasia is. Whoops. Okay, any questions from today or last time? I forgot to ask in the beginning. If you had any questions, you'd say, not from last time. Yes, sir.
You gave me a homework assignment.
Pardon?
I said you gave me a homework assignment last week.
Oh, yeah. Let's hear it. Homework. So I'll vote on it and give them a grade.
I couldn't find a lot of consensus on the exact number of copy number variants per person, especially defining copy number variants as 1,000 base pairs or more. But Eichler wrote in 2009 that looking at larger copy number variants, so it looks like on his array, it was only like 25,000 kilobases or more he was confident in. There's 3 to 7 on average per person. 5 to 10% individuals had a variant greater than 500 kilobases, and 1 to 2% had a variant greater than a megabase compared to the population. So the short answer, I think, is 3 to 7 for larger copy number variants, 20,000
Yes.
kilobases or more.
Right, that number is correct for larger copy number variants. And we talked about, we each have 6 million SNPs, that is subtle single base pair substitutions in our nuclei. Since that time that Eichler wrote that paper, there's a paper last year, I think, in the last 1 or 2 years, which I believe he also co-authored. And it was based on the T2T genome, if you all, right? And which is a much more accurate depiction of the human genome and the variation thereof. And in that paper, he was able to, he and his colleagues were able to look at smaller copy number changes and get down to roughly 1,000 base pairs, insertions or duplications. The median insertion is 2 versus 3 copies, and in some cases, it's always got to be 2 copies. One can lose a certain part and get down to 1 copy, but that's unusual. But the median size is 10,000 bases, and there are thousands of them. I don't remember how many there are per patient, but what grade do we give him for that report? Let's see. Alex.
Out of 100%.
100%, man, you're an easy grade.
No, he said out of 100%.
Out of 100%.
80.
I'll take a B minus.
All right, we'll give you a B minus, but with a chance to get an A plus. Okay, look at A plus. Look at Evan Eichler's more recent paper and email us. That means down to the TAs. Okay, and they will communicate it to everyone along with a couple of papers for you to read that I'll send them, and saying what the average number of copy number variations in a typical person is, and then we'll increase your grade to A plus, assuming you get the right answer, which I'm sure you will. All right. Any other questions? All right. How many genes are there that are driver genes? There are about a couple of 100. It depends on how you define driver genes. But the best way, as we discussed last time, is through genetic alterations, the frequency in cancers. About 2/3 are tumor suppressor genes, and the remaining third are oncogenes. Now, this is a very important distinction between oncogenes and tumor suppressor genes. You'll note that the 200 driver genes, there may be a few more that are uncommon, but together they're only 1% of the total human genes if we say there are 20,000 genes. There are key differences therapeutically between tumor suppressor genes and oncogenes. The distinguishing feature of oncogenes, one of them, is that they're activated. The accelerator is stuck to the floor. So you can drug them. That's the vernacular for treatment. If something is stuck, if it's overactive, you can design a small molecule compound that fits into the active site of the protein encoded by the gene and disarm that activation or disarm the function of that activated gene. So all of the so-called targeted therapies for cancer are based on targeting oncogenes. Now, tumor suppressor genes, the difference is they're inactivated. The brake is gone. You can't drug something that's inactive easily. If you think about drugs used in the clinic, it doesn't matter whether for cancer or anything else, they almost always either inactivate a protein, in some case they may otherwise change that active protein, but they need a protein to do something to. And with tumor suppressor genes, often the protein is gone from the cell. There's no protein. Because, for example, if there's a stop codon, nonsense-mediated decay, which occurs at the RNA level, will make sure that that RNA is degraded and no protein is produced. And even without nonsense-mediated decay, the protein may be truncated so that it's not functional. And it's almost impossible to reactivate a broken protein with a small molecule compound. So to date, there's not a single example of a drug for a tumor suppressor gene, despite lots of trying. For example, with p53, the special example, which we'll talk about later, that protein is still present in the cell. But it's not easy to turn it from inactive to active. So if you're going to target a tumor suppressor gene, and what would you do if you were a drug developer? How would you at least counterbalance the effects of the tumor suppressor gene if you can't develop a drug that actually turns it back on from its off state?
Supplement with the tumor suppressor gene or a similar functioning gene or upregulate.
Can you say it louder?
I would supplement, if it's P53, find a way to supplement it or upregulate a gene that does something very similar or a protein.
Yeah, you're on the right track. I mean, all these genes act in pathways. They don't act alone, right? So if a gene is turned off, it means another gene in the pathway has to be turned on, right? And if you have a gene turned on, that is suitable for drug development. You know, most of the targeted agents now in use are kinases. They add a phosphate group to proteins. They have a small binding pocket for the phosphate, for the ATP to bind to the protein. And you can get an inhibitor that binds to that pocket and keeps it from phosphorylating the protein. So perhaps a kinase downstream of the tumor suppressor gene is now aberrantly activated. And Daniel,
Yeah.
what is the problem or the disadvantage of targeting that pathway compared to targeting the mutant protein itself?
Regulating it at the right level, because you need some activity of that pathway for it to work in the normal cell.
That's precisely correct. If you target the drugs that target the mutant oncogene, for example, these kinase inhibitors, only inhibit the mutant protein. They fit into the active site of the mutant protein much better than they fit into the site of the normal protein. So they're much more specific. But if you target a pathway that's downstream of that mutation, you are going to also inhibit that protein in normal cells. And all of these genes have functions in normal cells, important functions. So you get more toxicity to normal tissues. This is called the therapeutic window, the difference in dose that's required to kill a cancer cell compared to a normal cell. And then you blur the distinction between conventional chemotherapeutic agents and targeted agents. You can still call them targeted, but it's kind of a misnomer. Conventional chemotherapeutic agents, which are still the mainstay of oncology. Right now, only a small fraction of cancer patients can be treated with targeted agents, and I'll explain why in a minute. But all cancer patients are generally, with advanced cancers at least, are treated with conventional chemotherapeutic agents. And these include microtubule inhibitors, alkylating agents, all kinds of intercalating agents. And they work. They don't induce cures in most cases, but they can induce remissions that are important. And now, why does a microtubule inhibitor or an alkylating agent, what is the therapeutic window there? Why does it kill cancer cells more than normal cells? Zoe, take a stab. Is that how you pronounce your name, Zoe?
Yes.
Take a stab. Why do they work at all?
Because the cancer cells are growing a lot faster than the normal cells.
That is the answer that is in the textbooks. And it's totally wrong. So, I had to grade that answer. What can I say? Oh, you read the textbooks. That's good. But it's wrong. Because self-renewing tissues proliferate, grow. I mean, it depends on how proliferation is defined. But they turn over as quickly as cancer cells. The difference between cancer cells and normal cells is not their rate of cell birth. It's the S. It's the difference between cell birth and cell death. So it's not simply because the cells are growing faster in a way. Faster has to be defined by dividing more. Some oncogenes and tumor suppressor genes result in decreased cell death. Others result in increased cell birth. It doesn't matter. They have the same net effect on S net cell growth. So, why do these conventional chemotherapeutic agents work at all? And they do. For example, they cure many cases of childhood leukemias. 80 to 90% cure. Totally conventional chemotherapy. Conventional chemotherapy are basically poisons. Some of them developed from chemical warfare dating back to World War I. But they kill cancer cells. Another one is radiation. That's a mainstay of cancer treatment. That's not targeted. You just irradiate the organ where the cancer is. So why do they kill cancer cells more than normal cells? I don't know. No one knows. I can speculate and wave my hands and say, well, they have disabled pathways, which for some hand-wavy reason makes them more susceptible to certain of these agents. But the truth is, nobody knows.
Do these treatments also work in people who are already immunocompromised when their cancer is identified and then before treatment begins?
That's an excellent question, which I could spend the rest of the hour on. So it is often believed that cancer results from a defect in immunity, which is related to your question about immunocompromise. So are cancer patients immunocompromised? What role does the immune system play? Well, you asked the question, so what do you think?
I would think yes, that the immune
Yeah,
system.
and you might think yes, and that was a popular view many years ago, and it's still a popular view in the lay press. You know, if you look at health supplements, you often see that this is whatever substance they're talking about activates the immune system, and that will prevent cancer is pretty much nonsense. So here's the evidence, so you can form your own conclusions. Let's take a patient who's unfortunate enough to have HIV, and this is before treatment, or even with partial treatment, or a patient with a genetic abnormality that makes them immunocompromised. Do they get more cancers? If they did, it would support your thesis, correct? And if they didn't, it would support the other view. So do they get more cancers? Anybody? Take a guess. It's either yes or no, so you got a 50-50 shot. Raise your hand if you think yes, they get more cancers. Okay, raise your hand if you think no. All right, well, you're all right. So, yes, they do get more cancers, but the key is they only get cancers which are virally induced, right? So they get lymphomas, which are induced by viruses, or other types of cancers that are induced by viruses, which means that the immune system, as is of course obvious when you think about it, was not destined to avoid cancer. It was destined to keep people from getting infections. So these people with poor immune systems, for whatever reason, are indeed susceptible to cancer, but largely to cancers of the immune that are caused by viruses. They do not have a higher incidence of the common cancers, say of the breast, the colon, the lung, etc. Now, just because the immune system does not protect people from getting non-virally induced cancers, it doesn't mean that you can't exploit the immune system to treat patients. And one of the most promising forms of therapy that has developed in the last decade for cancers is called immunotherapy. And the passive kinds of immunotherapy that is most effective is called immune checkpoint inhibitors. I'm sure some of you have heard of that, but some patients with cancers can be treated quite effectively, and some even cured with immune checkpoint inhibitors. And what these immune checkpoint inhibitors do is they allow immune cells, say T cells. I won't go into too much detail because a lot is speculative, but somehow they allow the immune system, which is inhibited, to become more active. So any antigens which are present on tumor cells then can be better recognized by the immune system in the presence of these immune checkpoint inhibitors and allow the immune system to shrink the tumors or eradicate them. Now, what are the likely side effects? This is a tough one. Janae? Did I get it right?
Yeah.
All right. I get an A+. Janae, what are the likely side effects of immune checkpoint inhibitors?
I'm guessing that it can, that they could possibly be non-specific, and so you might end up killing like normal, regular cells at some point.
And what diseases could that cause?
I'm not sure what type of disease.
All right, call
So like
for
autoimmune
help.
disease.
What?
Different autoimmune diseases.
Yes, you got it right. Autoimmunity, right? That's exactly the defect that patients have who have an immune system that functions too well, in a sense. It recognizes antigens that it shouldn't. And the main side effect, one of the main side effects of immune checkpoint inhibitors is autoimmune-like diseases, which often can be very distressing and even lethal. So we're up to Carter. So Carter, what kinds of tumors do you think? Let's continue because we'll try to continue. Maybe take a little break later. But thank you for reminding me. Carter, what kinds of tumors do you think these immune checkpoint inhibitors would work best in?
I think they'll work best in tumors that have like immune activity or able to have immune activity. I think I've read they talk about like hot tumors and cold tumors. But these are tumors that are able to be infiltrated by the immune system. And if it's like so big or if it's in some part of the body that doesn't really have immune cells like active, then it doesn't make much of a difference.
You're right. That's part of the answer. But go a little bit deeper. Why are some tumors hot, which means they have immune infiltrates, and others are cold, which means they have a lesser immune infiltration or composition of immune cells, like T cells, macrophages, etc. Why do you think? And I'll give you you think for a second, then I'll give you a hint. All right.
The first guess that I have is that if it was like a leukemia or something, if the white blood cells are the cells that are affected in the first place, it might not matter. You don't have immune system at all to fight your infection. The second one is, I was thinking about like vascularization, but the tumor is going to have its own vascularization anyways. I don't know beyond that.
Okay, those are reasonable guesses. Now the hint is this is a course on human genetics, and this lecture is cancer genetics. So does that give you a hint?
Is it they have mutations that are detected like through the MHC, like if they're not being detected
now you're on the right track.
if they don't have mutations that are like causing proteins that would be flagged by the immune system, then even if you like block PD-1 or whatever, the immune system won't recognize that cell as being bad anyway. That's not killing it at all.
Perfect. So what tumors do you think might have more mutations?
We already talked about solid tumors need more mutations.
No, but what tumor type? tumors of what organs, what kinds of tumors might have more mutation. Let's get you some help from your friend Alex.
Oh, Alex actually just tapped my hand, so I'm going to think melanomas, perhaps, because they're more
Perfect.
exposed to radiation.
Yes, you mean ultraviolet radiation.
Ultraviolet radiation.
Yeah, give me another.
I mean, maybe someone who has like a lung cancer that was induced by
Perfect.
smoking. The
Right.
ones that are caused by environmental insults.
Yeah, the main two are ultraviolet and smoking. You know, you can look at people who are working the radio lines, but they're not very common now. And there's one more. That's
Yeah, anything else?
particularly
What you got,
susceptible.
Alex?
I don't know, maybe gastro.
Yeah, it's a kind of gastrointestinal, but this is a story that is very dear to my heart because it comes from the collaborative work of Suzanne Tepalian. Is a professor here of surgery, but she did the first trial on immune checkpoint inhibitors with a PD-1 inhibitor, and showed that it worked only in lung and melanomas, okay? And didn't work in any other tumor type. And they had no idea why, because at that point, immunologists were not thinking like geneticists, right? They were only thinking about immune cell infiltrates and vascularization, you know, common things like that. But they visited our lab, and Drew happened to be the first graduate student I trained in our lab. I don't know if any of you know Drew, but he's a terrific immunologist. And they told us about these results, and we said, well, did anyone respond who didn't have lung or skin cancer? And Suzanne said, oh yeah, there was this one patient with colorectal cancer. She was a 56-year-old woman. She had a complete response, and she's cured. And this is now 4 years. This was before the paper was published, but they had treated her a few years earlier. And I thought of it, and the members of our group, I mean, we immediately had this kind of eureka moment. And we said, we know exactly why. That patient must have mismatch repair deficiency in her tumor. And we asked her if she did, and she said, I have no idea. So she went back and checked, and in fact, she did have a mismatch repair deficiency, which means she had roughly 1,500 to 2,000 mutations instead of 50 to 100. So just as you said, a lot more neoantigens to have the immune system react to. So we were really excited about this, and we immediately sent off a paper to the New England Journal, a little letter, you know, remarking on why patients with lung and tumor responded and predicting mismatch repair deficient. We thought this was amazing, and it just took a few days for that journal. Did I mention the journal's name? I shouldn't have, but it's a well-known medical journal based on the northeastern part of the United States. Didn't take them long to send us back and say this is totally speculative and not worth publishing. So that didn't deter us, and we set up a trial. We tried to convince pharmaceutical companies to give us some immune checkpoint inhibitors and underwrite the trial and pay for it, but they also thought this wasn't a very good idea. So Hopkins actually paid for the trial, which is extremely unusual. And one company was generous enough to give us the drug to use, but they wouldn't pay for the trial. At any rate, this resulted in miraculous results. Our first patient was actually a graduate student, one of our first patients, who was he was scheduled to go into hospice care on Friday. I remember it was a Wednesday when we got the result, and he was supposed to go into the hospice on Friday. So the study coordinator called him, asked him if he wanted to go in the trial, and he said, of course. And he took the drug, and this was like 2015, so it's 10 years ago. He's cured. He graduated, he has a family. So that was a very memorable experience, which is related to this course. But I think it emphasizes that when you understand genetics, you can explain a lot of stuff, even immunology. All right, where are we? So, oncogenes, here are ways to activate them. I think you're all aware of this. I'll go through it quickly. We discussed it a little bit before. You can activate them by mutation. They usually become, that is a single base substitution, an amino acid change in a part of the protein that regulates its function. That's one common way. Another way is amplification. Normally you have 2 copies of every gene in your cell, but sometimes through the amplification of a gene, a little part of the genome, it can be up to 100 or 200 copies. And then the protein is the same, but there are many more copies of it. So the signal that it transmit is exacerbated. A third is translocation. You can translocate part of one gene to another gene, which activates it. For example, you can translocate it to the promoter and enhancer region of another gene, which then would make it expressed much higher than it normally would be, and not expressed in a regularized way. So that's similar to amplification. The protein may be the same, but there's more of it. Or it can translocate, so now it's a fusion protein between another distinct protein and the oncogenic protein, which can endow it with new properties. And also there are viral oncogenes which are integrated into the cell through viral infections. They are not normal cellular genes, but they can play a major role in cancer. And this is the way that the Philadelphia chromosome through translocation is generated. It is through, there's chromosomes and genes. One, the Abel gene is the oncogene, and it gets translocated to another chromosome, which this translocated chromosome 9 is called the Philadelphia chromosome, simply because it was discovered by Peter Knoll in Philadelphia. But at the molecular genetic level, what this translocation does is it fuses 2 genes together, one on chromosome 9, one on chromosome 22, makes a new transcript, new fusion transcript, new fusion protein, and at the biochemical level, that alters the structure of the active site. The Abel gene is a kinase, so it phosphorylates its client proteins. You can develop, it has been developed, the original drug was called Gleevec, binds to that site, prevents binding to the ATP, prevents the phosphorylation of the client proteins, and bang, it can inhibit tumor cells from dividing in a very specific way. Again, this is a targeted agent, quite a bit different from conventional chemotherapeutic agents because it only targets the product of the mutant oncogene. The other way to activate oncogenes, in a sense, is through viral oncogenes. Now, there are several kinds of tumor viruses. Papilloma viruses, anyone knows what kinds of cancers these viruses cause? I'm sure you do. Yeah.
It's most common cervical cancer, but it can also affect like penile cancer and a couple of other.
Correct. Penile cancers, anal cancers, and most recently were recognized, at least recently, oral cancer. So it's sexually transmitted. Another kind is papoviruses. SV40. It's a long history here. Was worked on extensively by Dan Nathans, who won the Nobel Prize for his work on SV40 and restriction enzymes. And anyone knows what cancers these cause? In humans, not sure they cause any cancers, but they're good experimental tools. There is some evidence that the JC virus may cause some cancers. Adenoviruses, again, are largely experimental. These are cold viruses, but they do contain oncogenes, and you can transform cells to neoplastic in vitro or in animals. And the fourth, and this is a class that's important in cancer, is Epstein-Barr virus and HHV8, which stands for human herpes virus 8. EBV causes what kinds of tumors? Anyone know? Burkitt's lymphomas, which are common, relatively common childhood cancers. They're lymphomas that occur in Africa. HHV8 causes cancers in patients with AIDS. Kaposi sarcoma, again, emphasizing the role of virally induced carcinogen, virally induced cancers in patients with immune defects. Now, EBV causes another disease, which probably at least half of you, maybe more, have had. I'm not going to ask Maya because I'm sure she knows the answer to this. But she cheated and she went to medical school. But anyone else? No, EBV. Mono. Okay. Anyone had mono? You don't have to tell me, it's confidential. Many didn't have it. Some of you probably have had it. I mean, most people are infected with EBV. Some of them get the disease, others don't. And the disease, if none of you have had it, is associated with a sore throat, fever, and swollen lymph nodes. So the EBV infects B cells. It's a virus that infects B cells and causes them to proliferate. And you can see their swollen lymph nodes. I mean, it looks like a lymphoma. So here's a good question for any of you, all of you. Do those patients with EBV have cancer? We'll start around again. Well, we can start with Maya. Do those patients with mono really have a cancer?
I would say no.
Why?
Because it's like, it still has like qualities of like more normal cells and less cancer cells. Like it will proliferate, but it will like proliferate within its container to some degree, like within the lymph node. It won't necessarily like try to like metastasize or like really kind of go beyond its normal confines.
Yeah, that's a really tricky question. Okay, so in terms of invasion, when you talk about liquid tumors, that's really not a particularly useful concept because liquid tumors, like leukemias or lymphomas, they're all over the body to start with. They normally migrate, whereas epithelial cells of the colon or the breast, the pancreas, the liver, they stay put. So when they invade through the basement membrane, that's very meaningful and unnatural. But with liquid tumors, leukemias and lymphomas, they're already migrating. They're all already invasive. So, you know, what's invasion? What's the difference between a benign and a malignant tumor? And when you're talking about a liquid tumor, you know, it's kind of semantic. If you define it the way I did, in terms of the difference between a neoplastic tumor and a non-neoplastic tumor, it is a neoplasm because the cells are proliferating. Genetically, the oncogene, that is the genes within EBV, are conferring a selective growth advantage, which is why there are more cells than they shouldn't be. So it really is a neoplasm. The reason it doesn't progress, so you can look at it as an initiated tumor, a benign neoplastic tumor. And it disappears because the immune system eventually handles it. And again, patients who have immune deficiencies, either inherited or through AIDS, can get EBV-induced cancer. Now, the reason it's a benign tumor from a genetic perspective is that it only has one hit, one oncogenic alteration. And in order to get to a cancer that is the malignant stage, you need two. And in patients with Burkitt's lymphoma, there's a second hit that occurs. Hit is the vernacular for a mutation. Before mutations were known. And that mutation is a translocation between the MYC gene and some other gene. And the other gene is an immunoglobulin gene. So why would an immunoglobulin gene translocation, Lauren, result in increased expression of the MYC oncogene? A MYC is a standard kind of oncogene that is a transcription factor. What do you think?
By evasion of the immune system, if the immunoglobulin gene.
It's actually simpler. The immunoglobulin gene is very strongly active in B cells, right? So the immunoglobulin proteins, antibodies, are produced in B cells, and it's the most highly expressed gene in B cells. So now, if you translocate a MYC gene, an oncogene, onto the promoter and enhancer of an IgG gene, instead of getting a very small number of copies, or 0 copies, or a small number in B cells of MYC, you'll get thousands of copies per cell. So that's an example of activation of an oncogene by translocation. The MYC gene protein is normal. It's the same protein, but now instead of, in a B cell, having, say, 10 copies of the protein, you can have 1 million copies of MYC, which then binds to the nucleus, activates growth-promoting genes downstream. So that's how you get from, and that is also a good example of this evolutionary principle. Now, one of, again, the most, I think, interesting, or at least informative discoveries in the past decades is the knowledge of these oncogenes within viruses. So in HPV and papillomaviruses, there are 2 oncogenes, one E6 and one E7. The names, again, don't matter. There's one in the SV40, it's called the large T antigen. It actually has 2 different components, so it functions as 2 genes. It's a large gene, that's why it's called large T antigen. And in herpes viruses, there's at least 1, perhaps 2. But the cool thing is that it was discovered by several investigators in the 90s that what the E7 and E6 oncogenes of these viruses do is that they bind to and inactivate tumor suppressor genes. So the E6 binds to and inactivates the p53 gene, one of the most important tumor suppressor genes. And the E7 binds to and inactivates the RB gene protein. So these viruses, these tumor viruses, are so powerful initiators of neoplasia because they inactivate 2 genes at once. So it's like getting 2 genes inactivated by mutation at the same time when a cell gets infected by an HPV in the cervix or elsewhere. And similarly, amazingly, even though the viruses are completely unrelated, the SV40 virus, large T antigen, its protein also binds to and inactivates p53 and RB. And similarly, the 2 oncogenes in adenoviruses, E1A and E1B, also inactivate p53 and RB. So this is obviously not coincidental. It shows that these 2 genes in particular are critical for controlling cell growth. And for some reason, viruses have picked these out as the best ways for them. I mean, they're not doing their thing, they're hurt people, they're not bad, right? They're doing it because they want to replicate themselves, they're selfish. And somehow they've figured out throughout evolution that the best ways to get these cells replicating and to get more copies of the virus produced is to produce these proteins that alter with the performance, the normal function of these 2 tumor suppressor genes, which is really fascinating. I think we're going to talk about Wilms tumor, and I think I'll do this in the context of a little bit of a genetic counseling session. I'll just tell you what a Wilms tumor is. Wilms tumor is a tumor of the kidney. And here's a picture of it. It's on the right side of the kidney. Most of these children can be cured, about 9% of them by surgery, but some can't. It's a cancer, can metastasize. There's a man named Knudsen, who was a genetic epidemiologist, who recognized there are two forms of Wilms tumor, familial and non-familial. And the familial, that is, it occurs in families, there's 10,000 times incidence. And the familial form, patients often get multiple tumors, more than one in each kidney or two in the same kidney. In the familial form, they often occur earlier, instead of 4 to 6 years of age, 3 to 5. And in the familial form, the key observation he made was that only a small portion of the cells become cancerous. Now, we now understand all of these observations that Knudsen made. This was back in the 70s, before any genes were discovered. But I'll jump to the bottom line, which is here. So in a normal human cell, starts like this, and there's more than one Wilms tumor gene, but let's pretend there's one, and it's on chromosome 11p, and it's called a Wilms tumor 1 gene. Normal cells in a person who's not predisposed have two normal copies of the WT1 gene. One of them gets mutated somatically. Just on the course of cell division. Remember, cell division is mutagenic. But it still has one copy of the normal wild-type gene. So that cell is physiologically normal. It has no selective growth advantage. But what happens subsequently, as the initiating event in Wilms tumor, is that the other copy is lost. It can either be lost, most commonly by a whole chromosome loss. That's called a loss of heterozygosity, because all of the genes and all the polymorphic parts of one of the chromosomes is gone. It can be the whole chromosome or just the short arm of 11. Same effect, it only leaves one mutant copy, or it can be a second mutation in the other allele. Either way, that cell is left with no normal copies of the Wilms tumor suppressor gene. That break is gone. So when we're talking about mutations and the number of mutations required to get to tumors, to a cancer, remember that for tumor suppressor gene, it takes 2 genetic events, not 1. For oncogenes, it only takes 1. You just have to stick 1 accelerator if you have 2 in the car to the floor to make the car behave abnormally, out of control. But if a car has 2 brakes, the same identical brakes, so to speak, you have to get both of them broken to keep the cell from stopping, at least for that brake. And of course you have an emergency brake, but that's a different gene. Now, in the non-familial form, that first hit is somatic. It occurs either during embryogenesis or early childhood. And the second hit mutation is also somatic. But in the familial form, that first mutation is present in every cell of the body, including every cell in the kidney. So that patient's kidney has a head start on the neoplastic process. It only needs one more hit. And when that second hit occurs, that's the tumor initiation. It doesn't result in a Wilms tumor like in the picture I showed you, but it does initiate that tumor. it still will require another mutation in another gene to actually get to a clinically evident cancer, but it starts the process. And I will stop here, and for our last 12 minutes, divide the room into a genetic counseling session focused on, let's see, Haley, you're the mom. Pick out a husband. Anyone you want as long as they're in this room.
Oh, okay.
Go ahead.
Dina.
I'm the husband.
Okay, who?
Me.
Okay. Although this is politically incorrect, I prefer you pick out a male, and it's much easier.
Okay. I'll do Anthony.
So pick out a male, even though, you may not want to.
I'll do Anthony.
Anthony,
Yeah.
okay. And now you get to do something that you never do in real life. Pick out a child.
I'll choose Danyan. Our son Danyan.
All right, your son Daniel. And pick out another child.
Zoe.
All right. Zoe and Daniel are the children. Now, Daniel, you have unfortunately developed a Wilms tumor. Very sad. But fortunately it was caught early, and it was cut out. And you have to be careful, and do ultrasound evaluations of you every so often to make sure you don't develop another one. But you're going to be okay. But Zoe, you haven't been born yet. You're a fetus. And Haley and Anthony have now sought the advice of the best genetic counselors in the world to determine whether they should allow you to be born. Which is in a sense what they're doing. So the best genetic counselors in the world, who amongst you would like to volunteer to be that? I would say Alex and Carter, with assistance from Janae. Okay, we'll have to be quick about this. And now, Haley and Andy, I gotta remember who the parents are. Okay? You are coming to see these esteemed genetic counselors, and of course you bring someone with you. This will be the state of medicine in just a few years. Whenever you go to the doctor's office, you want to bring someone with you. Who do you want to bring? I'll tell you. You want to bring lawyers. Okay, soon everybody, when they go to the day, you might as well take the lawyer. I mean, the doctor's going to see the lawyer sooner or later. They're sooner or later going to make a mistake. So Maya is going to change her profession.
No,
Get a
add
JD
to
now.
the profession.
Add some more letters after.
Yeah.
He has to do all the doctors.
All right, and everyone else is also an attorney, a malpractice attorney. My brother is actually a malpractice attorney. And it's a good way to make a living. It really is. And whenever the genetic counselors make a mistake, your job is to say, lawsuit! All right, so we only have 10 minutes. But go ahead, Haley and Anthony. Well, the genetic counselors, introduce yourselves really quick and what you're going to do.
I'm Janae, and I will be assisting Carter and Alex.
And I'm Carter. We're going to be helping you understand the genetic context of your children today.
Yeah.
All right. In the interest of time, we'll go with that. And something I didn't tell you is that Haley is a mathematician, and Anthony is an atomic physicist, and they want to know the precise chances that their fetus, tentatively named Zoe, will develop a Wilms tumor. So genetic counselors, again, I'll shorten it a little bit, but they have a child with Wilms tumors. They have Daniel, right? You have a Wilms tumor. You're not in this section. You're in the outside of the room.
The babysitter's taking care of it.
Yes. All right, so ask them some questions.
Do either of you have anyone in your family who have had Wilms tumors or tumors like Daniel's, especially at a younger age?
No.
What'd you say?
No.
No. How about you, Haley?
No.
Yeah.
I don't-
No. Okay. So, what's your answer to that question? What's the chance that Zoe's going to develop a Wilms tumor? Based on that answer.
I'm scared of Maya over here, but I'm going to say it's very unlikely. If you have no one in your family, and we're pretty sure that Anthony is the father.
Well, that was insightful, because he may indeed not be.
How old is Anthony?
Wow,
Anthony, how old are
messed
you?
up.
Yeah.
How old are you?
Anthony, how old are you?
I'm 62.
Let me tell you, that's not old. It's all perspective. Am I going to sue the bastards or not?
I'm trying to think. So like obviously it depends on when the second hit occurs, but from what I remember of thinking more of like retinoblastoma, like it occurs. I don't want to say commonly, but like between siblings.
Yeah, but well, you didn't get their answers. It's not fair to sue them yet. So what are the chances of Zoe getting a Wilms tumor? Well, order of magnitude.
It seems like it's
Yeah, depends on the number.
Do we know where is the tumor? Is it in one kidney or in both kidneys?
Good question.
Oh.
Daniel, where is your
I'm too young to know that type of thing.
You're very precocious. You have smart parents.
One kidney.
One kidney. All right, give me an answer. Order of magnitude. 10 to the 6th, 10 to the 5th, 10 to the 4th, 10 to the 3rd, 1 in 1,000, 1 in 1,000,000.
Yeah.
Alex says 10 to the 6th.
10 to the 6th. Okay, lawyers, lawsuit or not?
It feels like it should be more.
Feels what?
It feels like it should be lower. I would think.
I could be, but they're going to get sued anyway because you didn't ask the right questions. Okay, like maybe they're orphans. They may not know their family history. So, very poor questioning from these genetic counselors. All right, so, but let's assume for the sake of argument that they're Amish and they know exactly their family history. Well, not Amish, that's too good. But, regular people.
Sure.
And they don't know of any history, family history. Okay, your next question.
How old were you when you first conceived Daniel? Haley and Anthony.
You said 60?
Yeah, 60.
Haley, how old are you in this situation?
He said 62.
40. No, 40.
You're 40?
40.
All right. I mean, that's what you're getting at is the older it is, the more likely that a mutation comes from any. But it's going to be hard to give a quantitative answer to that. So you can ask other questions. Well, suppose, let's put it this way. Suppose that's all the information you had. And suppose I was giving this course in 1991, which I did. Okay. And so, if it's a non-familial form, the chances are 1 in a million or something like that. I don't know exactly. But Wilms tumors are a rare disease. Suppose it was familial. What would be the chances that Zoe would get a Wilms tumor? So we have 1 in a million if it's non-familial. What are the chances of Zoe developing a tumor if it's familial? And attorneys, get ready.
What percentage of people who inherit the mutation develop the disease?
Great question. Okay, and the formal name for that concept is penetrance. Okay, so 90%.
Is this dominant?
Yeah.
50%.
But then the parents must have
And we're sure that they have no
10%
family,
of
no family history.
He just said assuming familial.
Oh, assuming it is familial though.
Yeah.
Assuming it's familial.
Well, then I think it would be 50% times 90%, so
Exactly
it would be
right.
45%.
45%, right? 90% times 50. Well, no, that's, there's one more thing, okay? How do you get to 50%?
We're assuming that one of the parents is carried.
Correct. So it's 50 times 90 is 45%. So here we have, so they come all this way. I mean, they live in New Guinea to see you guys. Okay? The world's best genetic counselors. And you can tell them in 1991 that the chance of Zoe developing Wilms tumor is either 45% or 1 in a million.
Yep, that's great.
That's it. That's all you can do. That was not worth the trip from New Guinea. All right, and I say that because this is how understanding of the genetics can help with genetic counseling. But we can do tests, and you have access to the world's best cancer genetics laboratory in the world, mine. So you can do any test you want. So what test do you want?
In 1991?
No, now. 25, March, whatever, April 2nd.
No, we should both.
First, who do you want to test?
So we want to do trio sequencing and we want to do NIPT sequencing.
Okay, well, their insurance, they live in New Guinea, they won't pay for that.
Oh.
We
Yeah,
deal
what
with
you
the
gonna do?
U.S., so you got to pick one person to test.
Define person.
One person?
One person.
Can't we just test the fetus, or they gotta be alive?
You can test the fetus. Would you like to do that? Lawyers? Lawyers? Lawsuit?
What do they want? What do they want to do?
Why is that a lawsuit?
Are they going to consider, like, is this them? They're considering aborting the fetus?
It's none of your business.
That's right, it's not.
Just get the dad's high. It's just blood test from mom.
All right, yeah.
No harm.
All right, test the mom. Lawsuit? Anybody? Why? Well, okay, mom tests negative. What does that mean? How does that alter it?
Well, we can alter the probability.
You can. So instead of 45%, or 1 in a million, you get down to 22.5%, or 1 in a million. So not the best person to test because it might come from the father. Okay? You can't test both. You could test the father and get the same result. So you want to test, who's the kid? Yeah, Daniel. Okay, we test Daniel. What tissue do you want to test?
The kidney tissue, right?
Well, I would test something that's not in the kidney.
That's not what?
That's not in the tumor. I would test not like normal tissue.
Okay.
And that would tell us whether it was a germline mutation or if he happened to have some somatic mutation in the tumor.
Okay. What tissue? Cheek bleed?
Well, they have one normal kidney. Can we test the normal kidney?
That's a good one.
Yeah.
Okay, yeah, again, in the interest of time, the optimum tissue to test is actually, this is a subtlety, is the tumor. Okay? And that'll become evident in a minute, but that's not what's generally tested. What's generally tested is the blood. Okay, because it's easy to get. Or, you know, you can test a mouthwash, whatever, you know, or cheek scrape. Okay, so you send that to the lab. My lab says no mutation in the WT1 gene. Now I report back to the parents, they come again from New Guinea or wherever they're from. Okay, and what's their chance of Zoe developing a Wilmster? Again, they're mathematicians. So you want exact, or at least the reasoning for your exact statement.
So we tested the blood, and the blood did not have WT1 mutation. So
Okay.
I think we're right.
So give them a number.
All right. So we have determined that we think it's. Did we test for any other mutations? Anything like
Nope,
that?
just
So
WT1
we
gene.
don't
That's
know
all you
like
paid
DNA
for.
damage repair or anything like that.
That doesn't cause Wilms tumors. Only the Wilms. We're pretending only the WT1 gene causes Wilms tumors.
There's no
And in
modifier
fact,
loci.
no, there actually aren't any
we'll say
modifiers.
we're gonna tell them that their next kid.
Tell them, they're right there.
All right, you guys. Your next kid has the same probability of developing a Wilms tumor as the general incidence of Wilms tumors in your population where you live.
Excellent answer. Lawyers?
I'm okay with it.
Lawsuit? What are you missing?
It's not ideal.
I know you know this. Okay? But you know, you send it to my lab. What did I test? I'm a standard lab, so I only test the exons of the WT1 gene. Okay?
I thought this was the best lab in the world.
That's all I can do. And even in the best lab in the world. Okay, suppose the whole gene is missing. Even if I do whole genome sequencing, I may not see that. Certainly if an exon, I may, but I may not. And clinically it's not done, it's just exons. It's too expensive to do a whole genome sequencing when you're only looking for a small gene like WT1. But at any rate, so you have to figure in the sensitivity of the test, right? So if the test is only 90% sensitive, so there's a false negative rate of 10%, then that alters the probability, right? And that's a conditional probability which you'd have to give them. It's still much less likely. It's 90% less likely. The other conditioning things. So now, if you're going to do this in true genetic counselors, you know, the really good ones, the way they do it is they can do conditional probabilities. So you know you have no family history. He has one tumor only. He has no detectable mutation in WT1. You do the conditional probabilities of all of that, and you get a probability to give them, which will be not 1 in a million, but certainly low. And that's what you would do. And then they're happy, and they decide to go have Zoe, and she develops into one of the world's best geneticists in the world, and makes an incredible contribution to science. We are in your debt, genetic counselors. Thank you.